The overall goal of the following experiment is to co-express different proteins in Escherichia coli and to assess their assembly in vivo. This is achieved by using the two compatible plasmids P bad and P good to co-transform E. coli top 10. As a second step, the co-transformed strain is cultured at 30 degrees Celsius and then induced to overexpress the proteins. Next, soluble proteins are extracted and subjected to gel filtration in order to assay the association of the overexpressed proteins. The results show that co-expression in E. coli is an efficient tool for the production of protein complexes assembled in vivo and is convenient to bypass the poor solubility of free subunits.
Protein co-expression is a powerful tool in biochemistry, letting the overexpression in single cells of different proteins. Going backwards, one can recognize that the overexpression of single genes was essential for the study of low copy number proteins. However, when the challenge was the study of protein complexes, one had to overexpress, isolate, and reconstitute in vitro single individual proteins. This is a labor-intensive approach, which also implies that the yield of the protein complex of interest is lower than those of the individual proteins assembled into the protein complex. In addition, one can face the problem that one of the protein interactors features low solubility.
This method can help answer key questions in the biochemical field.